my dear buddy Peggy Mason, Professor Peggy Mason, aka the Great One, professor of neurobiology at the University of Chicago, the host, curator of The Brain is So Cool. And I didn't realize that your MOOC, when I met you, Understanding the Brain, the Neurobiology of Everyday Life, has got a 4.9 out of 5 rating by students on Coursera. 4.5 out of 9.
4.9 out of 5.
9 out of 5. 4.9
Yeah.
out of 5. What'd you do wrong?
Yeah, I know. I gotta get those last.
that last 10%. I mean, my goodness, 4.9 out of 5 anyway. But I'm Aaron Freeman, artist in residence for the Chicago Council on Science and Technology. You are Peggy Mason. And you said you were having a good week.
I am having a good week. I'm out here in the wilds of British Columbia. My last week of my last chance to get away before December. I'm going in for a big haul there. Wait a minute.
I thought though that didn't the school year start?
Bizarrely, we are the latest. I mean, we are always late, but this year we are even later. We're usually the last week in September. University of Chicago usually starts the last week of September, and this year we are starting on October 2nd.
Well, I guess that's because of the Chicago letter that it's not a safe. Well, no, now it is a safe space for people who want an extended summer vacation.
Okay.
All right. Anyway, the fact that I remembered that,
How did you remember that, Aaron?
Well, I can't actually say because we're going to talk about memory, but not necessarily about human memories.
Okay, so great.
I have spent the time, but this is very confusing to me because you humiliated me some weeks ago when I was asking about. I was positing my rock solid theory, my rock solid hypothesis that memory was a function of the hippocampus, which you beat me over the head with, and it made me look like the nut job that I am.
Well, no. Memory is in great part dependent on the hippocampus. There's no question of that. But there are memories that are memories, and then there are memories, and then there are memories.
That's true. Now we're going to talk about how exercise can improve adult neurogenesis in adults, but before we do that, if you don't mind, because the study that we're talking about, which was published by our buddies in Neuroscience News, we love very much, and we recommend them to our Facebook and various.
Well, to be fair, the study was actually published in Science.
That's right. It was publicized.
I just want to shout out a little bit to Si-Hoon Choi, who graduated from the University of Chicago. He got his PhD from the University of Chicago. Very delightful student. He and a few others, Virginia Prevost and Mahesh Sharma, and I all are on Twitter, and we have a rainbow contest. Okay, so.
I said, how did I miss the rainbow contest? What is it now? You are neuro.
Well, it doesn't happen that often because a lot of time, months will go by and none of us will post anything because we haven't seen a rainbow. I was looking for one yesterday, but no. Anyway, so Si-Hoon was a great student. He graduated for a while ago from University of Chicago, and he went off to do a postdoc, and now he's an assistant professor in neurology at Harvard. And he's at Mass General Hospital, which is where I did my PhD work. So Massachusetts General Hospital in Boston. And he, the senior author on the paper is Rudolph Tanzi, Tanzi. But so what Si-Hoon did was to look at the effect of neurogenesis and exercise on a mouse model of Alzheimer's. And it's a really interesting result.
but it was not, well, yes. Okay, so what is the effect of exercise on neurogenesis in mice?
So we know that exercise increases adult hippocampal neurogenesis in mice. We also know
AHN.
What's that?
AHN.
AHN. I don't know how widely used that acronym is, but yes,
I saw it on the little thing. Okay, I saw it on the little thing, so that's what I did.
So exercise increases that, but the really interesting thing that they found was they could increase neurogenesis, or AHN, using other methods, either genetic or pharmacological. And remember, what was the result of that?
Well, it's very interesting because it worked. It did increase adult hippocampal neurogenesis.
But how did it work on well, okay, so how do we tell whether a rat is working on, firing on all pistons there?
That's a really interesting question. How do we know that? I don't know. Tell me, please.
So what we do is we put this radial arm maze. The mouse gets put in, so it's got maybe 8 arms. The mouse gets put into one arm, and 2 of the arms are baited, have food in them. And it's always the same 2 arms that have the food. So the mouse goes in, and on the 10th time that it does it, 5th time, whatever, it goes right to those 2 arms. But in this model of AD, the mouse goes to each arm looking for the bait. Okay. Not the swiftest boat in the race there.
So that's similar. That sounds similar to the water ledge test.
Yeah. The water.
The little
that's
island
used for how fast you give up.
Oh, I thought maybe the fact that some mice learn where the little kid.
There's that, but this is a good one. First of all, the water one is very unpleasant. There's a lot of wet water everywhere. It's very unpleasant for us. We have to do a lot of cleaning up, and the mice don't particularly like it. But this is okay. So they get they're not. I don't think they're food deprived. They get a bait. They can win this extra food or not. There's no big problem. And
if I can ask, please make sure that
2 arms, right? So you have 2 memories.
The idea is if the rat after the 7th or 8th try, the rat goes directly to where the arm where the food is, then we assume the rat has learned.
Yeah, the 2 arms where the food is. Yeah,
rat has learned.
This is a graded thing. So how long or how much what's the total path that the mouse goes? So if he walks 5 miles to get what could be a 10-foot prize, then he's not doing so well. He's not smart. He's not remembering things well.
And so in the experiments, they use a combination of pharmacology and gene therapy to increase the adult hippocampal neurogenesis in the mice, but it didn't actually make a difference in there. They didn't get. But how can this be? I mean, brand new neurons. Their little hippocampi are all like spiraling with brand new muscles. What happened? What went wrong?
Well, what's the first thing you do if let's say you're gonna make a new recipe? What's the first thing you do before you make the recipe?
Assemble the ingredients.
Yeah. Well, you clean your kitchen.
Clean your kitchen, yes. Clean your kitchen. On the good days, yes. Hopefully.
Clean your dishes. Clean your mixing bowls. Clean your utensils. Okay, everything's clean, so that what you add in, all the new stuff that you add in, is all okay.
Yep.
And so that didn't so much happen. So the thought is that when the animal exercises, not only does the animal make new neurons, but it also cleans up the environment so the neurons can actually survive.
Well, let me say, so what you're saying is that this business about generating adult hippocampal neurogenesis works when we know it works in mice who exercise, and it can also work in mice with just pharmaceuticals and gene therapy. But there's this other part, the setting of the stage wherein the neurons work. That part you cannot go. They could not in this experiment at Mass General. They could not replicate merely with pharmaceuticals and gene therapy.
Correct. That's exactly it. That's exactly it. So exercise, once again, you get for free what all these really smart people spend a lot of money and a lot of time figuring out how to do for a lot of cost. So what do? You want to live longer and live and have, be smarter, go out and exercise. Now, okay, before anyone freaks out on me, first of all, we have no evidence that this is going to work in humans. And there's some important differences between mice and humans. And second of all, many diseases, and I firmly believe this, even I suspect most, there's just this tiny little bit that you can influence it. And the time to influence it may be past. So most people that have Alzheimer's and get demented, there's nothing they can do at that point. There's nothing that they did 5 years ago. There's probably nothing they did 10 years ago or didn't do 10 years ago that sealed their fate. Their fate is probably sealed mostly by their genes, and then maybe by something that happened when they were 0 years old or 1 year old. Who knows? We don't know. You don't have a ton of control, but right. This is a very interesting thing that you can move the needle, or mice can move the needle a little bit by exercising.
So if you or I are going to develop Alzheimer's, there's a good chance that the plaques and the tau proteins that are going to misfire are already starting to do their thing in our brains at just such a level that, at least in your case, it's not so obvious.
You just don't know. And, right. And then, so Sihun, as I said, was a graduate from University of Chicago, and I found it really interesting. And I don't know, I tweeted something, and then he emailed me a few interesting things. First of all, he acknowledged that human and mouse adult neurogenesis are different beasts. We simply don't know what's going on in humans in the same way, and there's a lot of evidence against any substantial amount of neurogenesis in the human. The other thing that Sihun told me about, which was really, really interesting, is a group of people that have AD, Alzheimer's pathology, but they have no symptoms. Okay, so they're not demented.
What you mean by Alzheimer's pathology? You mean like the amyloid plaques?
The plaques and all that, but they are not demented. And
how can that be?
So what he says is these are rare individuals. They're cognitively intact at the time of their death, despite the presence of neuropathological features associated with a fully symptomatic stage of AD. Now, these people have an increase of adult neurogenesis or cells that stain as though they're recently born cells, compared to having the pathology with dementia. So it's possible that these incredibly rare people are being saved by some adult hippocampal neurogenesis, that they, for some reason, whether it's genetic or whether they're exercise fiends, who knows what's special about these rare individuals. But I thought that was really, really interesting.
So what do we know? Can we say anything about exercise and Alzheimer's in humans?
Exercise and Alzheimer. I mean, I think that what we can say is betting money would be on there being a protective effect of exercise. But how big that effect is and who it's going to work for and who it's not, those we don't know. We don't know. I mean, one other thing to keep in mind is that if you take away all adult hippocampal neurogenesis, and now we're back to the mouse. You say, okay, you, Mr. Mouse, don't get to make any hippocampal neurons in your adulthood. That animal never gets any pathology. It's a wild type. It's a regular old mouse who no longer does any adult hippocampal neurogenesis. That animal's fine. The animals that are affected by levels of hippocampal neurogenesis are animals that are susceptible. They're basically genetically engineered to get Alzheimer's.
This is an unpleasant marker, I think, because I think you're saying that that neurogenesis is a risk factor for the development of Alzheimer's, at least in mice.
Right.
I don't want that. I want neurogenesis. I want new brain cells, and I don't want them. I want new brain cells to diminish my risk, as do this rare bunch of people who are phenotypically Alzheimer's and do not clinically present. That's how I want it. Please, could you do something about that?
Well, do you know right now, Aaron, what days are we in? We are in the days of awe. There are 10 days of awe.
Of course, right?
And during the 10 days of awe, this is a Jewish tradition. It starts with Rosh Hashanah, and on Rosh Hashanah, God opens the book right. of life.
That's right.
And then sometime during those 10 days, in that book is written every person's fate for the next year. And on Yom Kippur, which is next week, the book gets closed.
And sealed.
And I think this is a very apt metaphor for life and neurological disease, which is that the book of life and the book of our fated diseases was probably written back when we were conceived, during gestation, when we inherited whatever genes we inherited from our parents. And that is a big chunk of what's going to happen. And our ability to influence is very, very small.
But now you bring up an interesting point. A buddy of mine named Sam Savage is a probabilist. So that when you say that our fates are sealed, our fates are largely written at birth, what you mean, I think you mean, is that the probability that we will develop Alzheimer's, develop whatever, is that probability is pretty well set. There are a lot of factors along the way.
It's anchored. I think a good way to put it is that it's anchored at birth, and it can move up and down. So, for example, there are a lot of conditions where you have a susceptibility, and then what you need to avoid are stressful life events. Okay, right. Good luck on that, but whatever. So you're supposed to go through life without any stress, and then your susceptibility is not going to actualize. But should you have a stressor, or maybe 2, or maybe 3, or 6, or 8, then the probability that susceptibility blossoms into a problem goes skyrocketing high. That's sort of the scenario that I think covers a lot of human conditions.
So, as a neurobiologist, to what extent do you actually focus on assessing probabilities of outcomes, of neurological outcomes in an individual over the course of their lifetime? How much training do you have to have in probabilistic neurobiology?
Oh, I mean, this is not my field. I have zero training in this. I'm
Okay.
I'm just reading the literature, and I'm telling you what my impressions are.
Yeah.
Yeah,
because it doesn't for what you do. It doesn't really matter because your job is to say, here's how this is what the chunks of the brain are, and this piece over here is that, and this one is connected to that, and this is doing that, and the probability.
I mean, I don't say that anything is uninteresting, and that you know, I used to think that there were uninteresting things, and when I wrote my textbook, I realized, yeah, not so much. Everything starts to be interesting once you look at it. There are always wrinkles there. I just haven't found anything that is not interesting anyway. So, and no piece of information is not useful. Pieces of information are useful.
So I want to say we're talking with we're the Chicago Brain Buddies, sponsored by the Chicago Society for Neuroscience. That there is Peggy Mason, professor of neurobiology at the University of Chicago. I'm Aaron Freeman, the funniest Negro comedian at the Chicago Council on Science and Technology. I got that nailed. So we are the Brain Buddies. We are generally here on Thursdays. If you have a question you'd like to ask, we have folks alive here. Loani is out running errands, and she may be in and out, so to speak. But if you have any questions about the aging brain and adult neurogenesis, don't hesitate to ask them as we go down here. But so does this particular research, again, well, it doesn't have any particular relevance to human beings, or does it? Are there things that we learn? Well, how generalizable can we say, like 50% generalized? There's no way to quantify the extent to which
we don't know, but I think it's really interesting, this what Sihun was telling me in this email about these individuals, these rare individuals who have Alzheimer's pathology and no symptoms. Okay, so that's really, really interesting, and it suggests that at a broad strokes that neurogenesis also is influencing humans, maybe at the edges, maybe differently from how it influences mice, but I have a hard time thinking that any principle that we learn from mice will not be somewhere, somehow, to some degree applicable to humans.
Well, it's certainly not hard to imagine that you need, as the Jews would say, the Shulchan Aruch. You need to set the table. You can have your nice little brand new neurons, but you need to set the table.
Oh, absolutely.
That's not hard to think about.
That makes total sense.
Right. It makes intuitive sense. But let me ask you this. How often does something make intuitive sense to you that's just wrong?
It happens. and yeah, those are very interesting situations. It's a problem with incredibly intriguing ideas, but usually those intriguing ideas are ideas that came up, or the ones that get shot down are ones that came up without any data. This is an idea this is an interpretation that comes from data. So, they have this disassociation between neurogenesis effect within or without outside of the context of exercise. So that's a really, and they know this bit about BDNF, this neurotrophic factor that gets activated or released during by exercise. So BDNF is something that could be involved in the cleanup issue. So I mean, this is not just sort of that's a cool idea, let me propose it with no data. This is actually a data-driven interpretation. So I suspect they're right. What I don't know, and I don't think they pretend to know, is to what degree it will be applicable to humans or human disease. We just don't know.
Is there
At the
any
same time, I just don't, okay, what would be the downside in exercising? I don't think anyone sees a downside as long as your musculoskeletal system can take it. Yeah, go for it.
Well, of course, as I like to point out to all my friends at the gym, there's always a great reason not to go to the gym. There are important TV shows to be watched, important breakfast foods to be consumed. There's always a great reason. But in theory, when I was reading the articles in the paper, that in theory, let's just say what you are, and people at my age, I'm 62, of course, as I can mostly talk about, this brain health thing is a big deal. Something I'm concerned with, and I do what I can to try to do it. So that in theory, we know that we can get neurogenesis with the gene therapies and the pharmaceuticals. If there was a way to get this BDNF, what does that stand for, by the way?
Brain-derived neurotrophic factor.
Brain-derived neurotrophic factor. If there was a way to...
You've heard of nerve growth factor?
Yes.
Nerve growth factor is out in the periphery, discovered by Rita Levi-Montalcini, got the Nobel Prize for it. But brain-derived BDNF is sort of the brain mega growth factor. So it's sort of the brain's version of NGF.
And that's the thing that exercise produces in mice that has not been produced or created by pharmaceuticals or the gene therapy.
Correct.
So, but in theory, if we could get...
Well, that's one of the things. I'm not sure I don't know that BDNF is the only extra factor.
But that was one identifiable fact, one identifiable difference between the exercising mice and just the pharmaceutical mice. Right. So that in theory, is my point, that if you could find a way to do the BDNF and the other neurogenetic properties.
Well, the other factors. I mean, I think that's exactly where Sihoon and Rudy Tanzi are going. They're going to try and figure out what it is. And now they have this beautiful model. They can figure out what it is that exercise is doing and identify those exact factors, and then look at the effect of those factors, that pharmacopoeia, on humans.
So in theory, you could derive the same neurological benefits from without exercising. You could be neurologically 6-day-a-week workout person while you sit home, neurologically speaking. I mean, your muscles may atrophy and sarcopenia may set in, but neurologically, you could conceivably do it. And at which point, I wonder, if what you're concerned is your brain, why bother working out? You could imagine.
Well, you could do it in 100 years.
Well, there's that.
really not this year, and really not next year. And the way the FDA approval system works, really not in 5 years. So maybe, 10, 20 years from now, you got a shot at it, but we're not there yet. So you can't.
We haven't done anything like this. Is there anything like this, as far as you know, been any tests like this been done in humans?
Oh, there have been tests for various treatments of Alzheimer's, and they pretty much all crashed and burned.
But what about this exercise stuff?
I don't know of anything, but that doesn't mean much that I don't know it. So I don't know.
Once that it's.
I doubt it. I seriously doubt it.
Because
it's too new and it's too mousy.
So tell me why it is, if we absolutely positively know that mouse, as somebody said to me, if you're a mouse with cancer, we can do a lot for you. Right. There are many maladies that if you are a mouse, we can really help you out. But I want to just make sure that we don't totally lose our faith in mouse-based research and rodent-based research, because because that's.
I mean, I think mice tell us. I still believe that mice tell us a lot about fundamental processes. We just have to understand that those fundamental processes are going to be tweaked in us because of species-specific issues, and they're going to be combined in different amounts and with different emphases. The players are going to be the same, and we're going to learn so much more about the players from working on mice than we could ever learn in the slow process that it takes us to work on humans. So no question that this is a valuable way to go. It's just, it's the translation of it into humans is not the easiest thing in the world. But right now, before we go, I want to address Mike's question. So what about exercising your brain, math, puzzles, and chess? And I think that the answer has been that there's no evidence that the so-called exercise of your brain helps. I don't think that there's evidence, but I'm not sure.
I'm reasonably certain because Michael and I are both old Catholic school guys. He may well be referring to the very famous Minnesota nun study. Like a decade and a half.
So I think that, so for example, Luminosity tried to make the case that, if you do these puzzles, you're going to get smarter and you're going to blah, blah, blah. Well, the fact is that Luminosity's puzzles, you get better at Luminosity's puzzles, but you can't generalize them. And they have had to pay out for making that false claim. So there is no generalization of, say, exercising your brain every day on this puzzle. It's not going to help you solve a different type of puzzle. So I am not aware that there's any evidence that exercising your brain helps. So, there is also, you have to remember that in the nun study, it can be just simply be a correlation. It could be that the people that end up being more cerebral are the people that don't end up getting Alzheimer's. The number of exceptions to that is just myriad. So I don't think there's a lot of evidence for that, if any. As far as why do some Alzheimer's sufferers go between being lucid and not, the way I think about this, and this is not specific to Alzheimer's, but it's with any brain dysfunction, what really breaks down first are the long highways, the long connections.
As Sharon reminded us, the microtubules.
Yeah, so these are the longest pathways, have the most opportunity to break down. They do break down. Now there's traffic jams, so you can't use, for a Chicago metaphor, you can't use the Kennedy Expressway to get from my house to Aaron's house, but you can still do it getting through side streets. And so, you may get there, but you got there slower. And as to why it's better on one day and less, worse on a different day, I think these are just probably differences in amount of stress, amount of inflammation, the number of traffic jams that you have on your long highways of your brain, and as long as as the disease is not completely consuming and completely screwed up your entire brain, you're going to be able to have good days and bad days, and to get around those traffic jams and find other routes that will take you from here to there. But it's going to be, you're going to be oscillating around a lower and lower functional level. And that's the real tragedy of these neurodegenerative diseases, is that they are neurodegenerative. They go down. The neurons die. They just do. Sad and true.
All right. Well, so as we are the Chicago Brain Buddies, we always like to end on an optimistic, positive note, which I will look to my buddy Peggy is considering. Let me just say that I am Aaron Freeman. You're thinking about the positive takeaway from this.
I'm exercising.
Oh, no, you're exercising. I'm sorry. I missed it. You're right. You're exercising. Okay, so we are, but while Peggy is exercising and thinking about the great happy ending for this, we are the Chicago Brain Buddies. We're generally here on Thursday afternoons around 3 o'clock. Peggy Mason is professor of neurobiology. We encourage you to check us out on Facebook. Chicago Brain Buddies on Facebook. We also have a podcast, Chicago Brain Buddies, which is available on Stitcher, iTunes, various of your favorite Spotify, various of your favorite podcast outlets. And we are on YouTube at ChicagoBrainBuddies.com and the blog. So, so we thank you so much for your questions. We thank you so much for watching now. Thank you so much for watching later on. But Peggy, as we chill out here, give us from this wonderful new interesting experiment about adult neurogenesis in mice. What can we take away that's going to make our day?
Go exercise out in nature.
Exercise out in
you're gonna get two things. You're gonna get exercise, which really can't be bad from a cardiovascular point of view, obviously good. From a neurological point of view, probably really good too. And then being out in nature, there's a lot of growing evidence that this helps with mood disorders, and it helps with just mood in general, mood motivation, and yeah. So I would say, go out and do it.
So go out and do it. Contrary to American habit, get outside, get off your computer, go outside and exercise. Can't beat that, Professor Peggy Mason.
And do it with others. Socialize too. Go outside in nature and exercise in a social manner. You'll be hitting it.
Well, so you're saying that because you're up there in British Columbia where it's really beautiful.
And I have family, and we're hanging out, and we're taking hikes, etc. Yes.
All right. Well, thank you so much, Peggy Mason. We love you immensely much, and we will hopefully see you next week, where we will have. Actually, it was really fast. I don't know if you've seen that there's a new report out where they're implanting artificial lab-produced
brains,
lab-produced brains inside living brains, which is just really weird. So maybe we can chat about that next week. All right. Thank you very much. We are the Brain Buddies. See you guys really soon.